Well, welcome everyone to our second session of the Antarctic Seafloor, Ecosystem Interactions and Environmental Drivers of Change. Great to see you all here. On behalf of myself, Alix Post, and Narissa Bax and Hugh Griffiths, we're really glad to have you presenting and watching this session. So first up, I'd like to welcome Carlos Angulo-Prekler, and the title of their talk is Sponges, Good Natural Sentinels for Marine Biodiversity Monitoring, Fish Diversity in Antarctic Shallow Waters. Thanks, Carlos.
Hello, thank you very much. I'm going to try to share the screen. Can you see my screen?
Yes, that's come up well. Thanks, Carlos.
Okay, perfect. So I'm going to start. Good morning, or good evening, or good afternoon, depends on the place where are you. I'm Carlos Angulo-Prekler, and I want to thank to the organizer to allow me to present my work here in this session. I'm going to talk about the fish diversity in the Antarctic nearshore waters, and if we can use the sponges as natural sentinel for marine diversity biomonitoring. So the Southern Ocean encompasses about 10% of the world oceans, but it has shown a low fish diversity compared to other self-habitats. First of all, when we talk about the environmental DNA, we talk about a complex mixture of genomic DNA from many different organisms found in an environmental sample. It can be soil, air, or water sample. The ocean is a soup of its resident species genetic material. The marine eDNA suspended in seawater might contain living cells, metabolic waste, part of different organisms, or dissolved material. So the eDNA is the genetic material released from an organism into the environment. Commonly, the environmental DNA samples, such as water, are collected by filtering seawater through a Sterivex filter. However, in 2019, Mariani and collaborator came with a brilliant idea. They proposed the use of sponges as natural samples for aquatic biodiversity, based on that the sponges are the most efficient water filters of the planet, and they are constantly filter water from the environment. Our goals were first, validate the usefulness of natural sample DNA recovered from sponges to reveal the fish biodiversity patterns. We want to assess the efficiency in capturing DNA between water and sponge samples, between the different sponge species, and between the different pore size filters. Finally, all this previous methodological work is to describe the fish community on Antarctic shallow nearshore waters. To do so, we use sponges and water samples previously collected for a sponge microbiome study performed by Sacristan Soriano and College in 2020. During the austral summer of 2016, we sampled 4 different locations, 2 in the Southern Island, Deception Island and Half Moon Island, and another 2 in the Antarctic Peninsula, in the vicinities of O'Higgins Station and the Rodera Station. So we collected several biological replicates, between 2 and 4 replicates per sponge and station, of 4 different sponge species: the Dendrilla antarctica, Sphaerotylus antarcticus, Hemigelius pilosus, and Micalia serrata. These sponges are the most common and abundant in these localities. We also collected 3 water samples adjacent to the sponges for every location. So seawater samples were sequentially paced through polycarbonate filters of 2 different pore sizes, 5 and 0.22 micras. So in total, we have 60 samples, 36 sponges, and 24 water samples. Furthermore, due to the high stochasticity of the metabarcoding techniques, we also performed 3 PCR replicates for each biological sample. So what is called the technical replicates. The main methodology consisted on extracting the DNA from sponge tissues and then amplifying them using the specific MyFish primer for vertebrates, and then running the Illumina MySeq sequencing. And after running the metabarcoding pipeline, we obtained what is known as the molecular operational taxonomic units, the MOTUS, which are correspond with the different species. So moving to the results, here you can see the fish richness that we recovered from our samples. We obtained 15 vertebrate MOTUS from 183 PCR replicates. So 14 Antarctic fish and 1 marine mammal, the Antarctic fur seal. In order to be conservative, samples with less than 10 reads were omitted from the analysis. Although there is a great variability in the number of reads per sample, the species accumulation curve showed that we have enough samples to detect the diversity. Also, significant differences were observed in the number of MOTUS recovered by sponge and water samples, while no difference were observed between sponge species or between the different localities. On the other hand, both pore side filters also showed significant differences, but only in the species richness. Similar as happened within the sponge species and localities, the high variability between the samples resulted in no difference between the remaining diversity indices measured, the Simpson and the Shannon diversity index. The sequencing depth also showed a high correlation between the MOTUS detection and the number of total reads, with a greater efficiency for the water samples over the sponge samples. Here, orange dots represent the sponge samples and gray dots represent the water samples. As you can see, there is a predominancy of the gray dots in the upper right corner of the plot. However, the high difference in the number of reads between the different replicates suggests an important stochastic component of the PCR amplification. Despite the low number of total MOTUS detected in the entire dataset, each sample recovers several MOTUS, as you can see in the right side plot, with a maximum of 12 MOTUS per single sample. However, if we look at the ordination of fish community based on relative abundant data, in the MDS plot, we can see that there is no significant difference between the water and sponge samples in the left plot, while some difference were found between the different localities. For this reason, to study the fish community, we grouped both sponge and water samples and treated as one. If we look at the present absence data instead, if you look in the present absence data instead, you can see in the heat map where the color represents the number of PCR detection per each MOTUS at each sample. So to make it more clear, here in the y-axis, here in the y-axis, you have the samples, water and sponge, with the number of detection of each sample in the right side. So that means how many fish are detected in each sample. As you can see, it's slightly higher the number of detection in some water samples than in the sponges. The color of the bar represents the location where the sponges were collected. In the x-axis, you have the fish species detected with the number of total detection for each MOTUS in the upper side. The complementary bubble plot helped to identify the most abundant species per locality, using the total reads as a proxy for abundance or for biomass. As you can see with these two plots, Nototenia coriceps, here in the bubble plots and here in the heat map, what is the black rock cut, is the most detected and most abundant in all the localities studied. This species has been detected in more than 40 samples, and most of the MOTUS were identified at the species level, with only 2 MOTUS that were identified only at genus level, with a high taxonomical resolution. We found 6 different families where Nototenidae, with 8 species, represent more than 90% of the total reads recovered. The high dominancy of this family is something that is very common for Antarctic waters. we detected a fish core community shared among all the localities, composed by 5 different species, with only 3 species were exclusive to just one location. Deception Island, the locality with less species recovered, was also the most different one, with 2 species found only here. So beside the fish community, we recovered several reefs of the Antarctic fur seal, but only in Half Moon Island. Although we have not found significant difference between the different sponge species, each species were able to detect different fish species, while the water samples were able to detect all the species for the datasets, even some species that any sponge were able to detect. So to conclude, we can say that sponges are promising natural samples, able to detect the local ichthyofauna and differences between location, but they are not improving the results obtaining from the water samples, especially if we take into account that the water samples are only 3 replicates. Instead, we need several species of sponges with their own replicates to obtain a similar coverage. The sponge species do not impact the efficiency in capturing the DNA. So this methodology allows us to study the Antarctic fish community in shallow nearshore waters, overcoming most of the logistic constraints typically of the Antarctic waters. And finally, in line with most of the Antarctic studies, we found that the family Nototenidae dominate both the diversity and the biomass of the Antarctic fish community along the nearshore waters of the Western Antarctic Peninsula. Thank you very much. And if you have any questions, I'm glad to try to answer it. Thank you. I don't know if is everyone there, anyone there?
Yes, we can still hear you, but I think we lost Alix, who's convening. She's coming back now.
Okay, I was scared that nobody was hearing me.
Sorry about that. I lost my internet connection for 5 minutes of your talk, Cesar.
Don't worry, Alix.
Have you had a chance to have any questions?
Not yet, because I just finished it right now, and I was waiting if somebody are there or not.
Excellent. Well, if people want to put any questions in the Q&A box, or any of the panelists that are able to unmute can just ask a question directly. I'm not sure if you covered it in your talk, but did you get any sense as to any of what was driving differences in the fish communities?
No, because the next step will be try to measure some environmental parameters. But for now, our focus was to try to test if this methodology is useful and it will be possible to retrieve something interesting. Now, after we have found that there is a lot of chance and a lot of possibilities, and we have several ideas to improve the methodology, we will try to looking for the environmental parameters to try to differentiate why there are some difference along the Antarctic Peninsula.
Excellent. I'd really love to know more about the technique. I'll watch the recording to catch up on what I missed.
Sure.
But thanks, Carlos.
Thanks to you.
If there's no other questions, I'll just... But right now we have a lightning talk by Gabriel Lube on the spatial and temporal variations on sedimentary biogenic sterols of Mata Inlet, Admiralty Bay, Antarctica between 2002 and 2019. And I believe that that is a pre-recorded talk. So hopefully the IT team have that ready to go.
Hello everyone, I hope you're all well. My name is Gabriele Veralubi and I'm undergraduate student at the Federal University of Paraná here in Brazil. And today I come to tell you a little about my course conclusion work that is part of the Brazilian Antarctic project. My research seeks to evaluate the distribution and the source of biogenic sterol in Adamantium Bay, Antarctica during the years of 2002 to 2019. In this work, we choose 4 different sterols for natural organic matter, where the difference in numbers of carbons is related to specific sources of organic matter. Cholesterol was the predominant sterol, which was expected since, in addition to marine sources, there is a significant contribution for feces of several Antarctic marine organisms. Important contributions for citosterol were also observed, suggesting that there is input of organic matter from algae, diatomics, and cyanobacteria. Sterols such as brassicsterol and stigmasterol were also found with relatively high concentration values. The relevance of this work is based on the fact that the data obtained cover up a significant time series for studies in Antarctica. Thus, the results presented here allow a more accurate assessment of the viability of natural source of organic matter. In addition, knowledge of the characteristics and natural phenomena that involve sedimentary organic matter in Antarctica can clarify issues of global relevance, since this region is considered one of the main controllers of the global climate system. Thanks for listening.
Great, thank you, IT team. If you want to have a look at Gabrielle's poster, then please have a look at that in the e-poster section online. Next up, we have Paulo Camara and another lightning talk on non-fungal eukaryotic DNA present in South Shetland Island, continental margin, and Drake Passage sediments assessed using metabarcoding. Is there a recording for that one, hopefully?
No, we don't have any recorded video for Paul Komara.
Okay.
And Paul Komara also not joined the webinar. Has not joined.
Okay.
Okay.
All right. Well,
we can move for the next video.
Yep. So we can move ahead to Stefano Ambrosio.
Yeah, sure.
Have you got a recording for Stefano?
Yes, I have got this video for the Stefan.
Excellent. So that's going to be on comparison of sampling methods to assess marine benthic communities, visual versus destructive methods. Thank you.
So, welcome everybody to my oral communication in this 10th SCAR Open Science Conference. Today we are talking about a comparison between visual and invasive methods to assess the marine benthic communities. So the invasive methods like trawls, sledge and dredge were historically the most common methods of sampling the shelf benthic marine communities in Antarctica. While these methods are excellent for species identification of sessile and also slow-moving benthic organisms, they are not so efficient at capturing mobile species. Also, they are destructive and they cannot describe the species behavior and the interaction between the species and the ecosystems. On the other side, advances in underwater image technology, like for example photographic and video imagery, scuba diving, also the use of remotely operated vehicles and autonomous underwater vehicles have increased the knowledge about the demographic processes, structure, and also about the spatial distribution patterns of Antarctic benthic community, the Antarctic megafauna of the Antarctic continental shelf. Also, these visual methods, with these visual methods, we can sample a larger area without any impact on the bottom, and also are used to do behavioral studies of the species. On the other side, they are more expensive. They need also qualified technician to use it because they are difficult to use, and mostly they depends on the sea ice condition and the sea conditions. One of the invasive sampling methods used to access, for example, the in-fauna is the multi-box scorer. On the other side, to sample the sediment, the scientific community is using the multicorer. And most of the time, these two sampling methods are used together to see which is the relation between the type of the sediment and the in-fauna living in the sampling area. Also, he used the agassiz troll, but to sampling the megafauna and the epifauna. On the other side, the visual methods also are different between each other. The ROV are one of the most used methods. Also, the photographic systems are used to observe the seafloor. They are quite cheaper than the ROVs, and at the end, the use of submarine is a more sophisticated methods and also more expensive. In this study, the two sampling methods we compared are the agassiz troll and the ocean floor observation system, also known as OFOS, and both methods are used to sample the megafauna. The aim of the expedition was to evaluate quantitatively the assemblage structure, using also a standardized protocol that was used in the expeditions, some expedition before, and is a standardized protocol that is time and cost efficient. Also, this study has been done with the collaboration of many taxonomies that were present on board. We have to make a point that separately the data of the OFOS and the AGT has been published already by Good and collaborator in 2019-2016, but there were missing still a comparison between both methods. So the objective of this study were to compare the performance of two sampling gears, the agassiz trolls and the OFOS. Also to identify which classes are better sampled by both methods, and also to provide new quantitative data on the combination of different sampling methods on Antarctic megabenthos. The area, the sampling area is the Antarctic Peninsula, three different areas, the Drake Passage, with oceanic conditions, the Weddell Sea with continental condition, and then Bransfield Strait that is a mix of between the condition of the Weddell Sea and the Drake Passage. 14 stations were sampled in these three different areas, and also in two different depths: shallow from 100 to 350, and deep from 400 to 600. Regarding the OFOS, we analyzed 25 pictures for each station. In each picture, there were present three laser dots that make possible that there is a scale in the image. We analyzed for each picture 1 meter square, and in total we were able to count more than 10,000 individuals that were divided in 19 taxonomic groups. Regarding the AGT data, the data were the sampling methods was used with a speed of 1 knot for 10 minutes in the bottom, and for each sample we extracted a subsample of 50 liter, and only this subsample was analyzed, and we were able to count for more than 4,000 individuals, also coming from 19 taxonomic groups. This is the heat map we made for both sampling methods, and you can see that the most abundant species were for AGT the Polychaeta, and for OFOS the Ophiuroidea, but Ophiuroidea, Acidiacea, Bryozoa and Polychaeta were the most classes sampled by both sampling methods. Regarding the correspondence analysis, well, first of all, it was significantly for both factors, the factor area and the factor depth. Regarding the area, Alcionacea, Pennatulacea and Pycnogonida were a good describer of Drake passage for both methods, while for both methods Pterobranchia, Demosponge and Echinoidea were the representatives of the Weddell Sea. Regarding the depth, there was a bit discrepancies between the groups that were sampled. As you can see, the Scleractinian, the Polychaeta and the Gorgonian from the AGT are different from the Holoturia, Alcionacea and Bivalvia that are present in OFOS. Regarding the coinertia analysis, also we see a good separation between area. There was only one station that showed some discrepancies. Also there is congruence between taxonomic groups, as you can see from AGT, Crinoidea, Asteroidea and Holoturidea are in the same position of the OFOS in the graphic below. There are discrepancies between other species like Polychaeta, Scleractinia and Acidiacea. And well, this is a good comparison between the both methods. Regarding the background, there were only 2 studies that compared different sampling methods, one of Pineda Metz and one of Pippenburg. So there was a lack of comparative studies using different gear. From the results that we can see in our study, we can assess that visual techniques are highly recommended, and because we saw that for most of the classes, the two sampling methods they are not so different. So why not? We have to use more visual techniques to not have a big effort in the bottom of Antarctic communities. And thank you for listening.
Great. Well, that was really fascinating talk from Stefano. If you want to follow up with him, then please send him an email with any questions. IT team, can you please confirm that you have a recording for Simon Morley, who's up next?
Yes, we have the recording for the Simon Morley videos.
Excellent. So he's going to be presenting a talk on benthic biodiversity, carbon storage, and the potential for increasing negative feedbacks on climate change in shallow waters of the Antarctic Peninsula. Thank you.
Sorry for the inconvenience. Give us a 2 minutes of time. Sir.
Okay, thanks.
Hello all. Thank you to the Scientific Committee of Antarctic Research, SCAR, and India for hosting this conference, and to Narissa, Hugh, and Alix for convening this session on the Antarctic seafloor, ecosystem interactions, and environmental drivers of change. Firstly, I'd like to introduce myself. I'm Dr. Terry Suster, and I'm a polar benthic ecologist based at the Arctic University in Tromsø, Norway, and with an honorary fellowship through the British Antarctic Survey. You may have been expecting Dr. Simon Morley to be presenting this work as per the schedule, but he is currently in the Falkland Islands doing field work, and the internet bandwidth can be tricky. Simon and I are first authors on this paper published this year in NDPI Biology, but it really is a team effort from work which was carried out since 2013 at the British station Rothera on the Antarctic Peninsula. This work started with Belinda Voss looking at biodiversity and biomass of soft sediment habitats, and then during my PhD, biodiversity of Antarctic hard rock communities, biomass and energy use, which was submitted in 2017. In the same year, Barnes and Sands published a paper looking at functional diversity being key to Southern Ocean benthic carbon pathways. And in their manuscript, they pointed out that frequent ice scour may have disguised how highly productive Antarctic shallows may be, with the potential to immobilize a lot of zoobenthic carbon. I will explain in more detail what we mean by zoobenthic blue carbon shortly. Our paper here estimates the quantity of zoobenthic blue carbon found within both rocky and soft sediment, shallow seafloor communities along the Antarctic Peninsula, so coastal seafloor communities. It also looks at how this ecosystem service could increase with continued climate warming and potentially act as a negative feedback on climate change. As we're all aware of by now, anthropogenic, so human-induced carbon dioxide has been and is being released into our atmosphere and creating this climate emergency situation we find ourselves in. We therefore need to reduce the amount of carbon in our atmosphere, and the ocean acts as a large carbon sink, absorbing significant amount of carbon dioxide
through photosynthesis by marine plants such as phytoplankton. Carbon stored within trees on land is termed green carbon, whereas carbon stored within the ocean is termed blue carbon. And zoo-benthic means animals that live on the sea floor. Many people, when you talk about blue carbon, will think of kelp forests, seagrass bed, mangrove forests, etc. And these habitats are vitally important in their storage of carbon. However, they are also in severe decline globally. Living organisms, and in this study we're focusing on benthos, so benthic animals that live on the seafloor, they build their bodies from absorbing carbon dioxide from their environment. I will explain more shortly. But this can offer a nature-based solution to climate change by mitigating the amount of carbon dioxide in our atmosphere. Carbon is stored within their skeletons. However, if these bodies are buried, then the carbon is removed from the carbon cycle, and we say the carbon is sequestered. The importance of cold water blue carbon as biological carbon pumps that sequester carbon into ocean sediments is now being realized. Okay, so to explain zoo-benthic blue carbon storage, I'll use this figure, which was taken from a publication by one of our conveners, Dr. Narissa et al. 2021, and summarizes nicely how carbon removal from our atmosphere occurs and ends up stored in benthos, in benthic organisms. So if we start on the top left, carbon is captured by phytoplankton through photosynthesis and held for hours to months. As this captured carbon is consumed through the food web, it becomes stored in body tissues for months to years. Carbon is immobilized in shells and skeletons of marine animals for years to centuries. So here we're looking at the benthic organisms on the seafloor. When this is buried below the aerobic surface layers of sediment, it is sequestered for centuries to millennia. Sequestration is key, as this carbon is permanently removed from the carbon cycle. So as a recap, our paper looked at carbon storage within soft sediments and hard rock communities with the potential to becoming sequestered. Remember, the Antarctic Peninsula is a hotspot for climate change. There is research showing that with sea ice decline, as we see on the Antarctic Peninsula, there is longer duration of phytoplankton blooms, which will mean a longer feeding period for benthic animals, and therefore more growth and more carbon storage. So while habitats such as kelp forests and seagrass beds are in decline, polar benthic marine ecosystems could be absorbing more carbon and colonizing newly ice-free areas. As I mentioned, our research was focused around the British Antarctic Survey Research Station, Rotherham, which is situated on the west side of Antarctica, below South America on the Antarctic Peninsula, as you can see in this bottom left figure. The soft sediment fieldwork was completed by marine biologist Belinda Vorse in Hanger Cove, up here in the north, and South Cove, just to the south. And the hard rock fieldwork was completed by myself at Cheshire Island, just here, south of Rotherham Point. The first stage was to quantify the organic and inorganic blue carbon at 20-meter depth, which was done by drying the organisms at 60 degrees for 24 hours to obtain dry weight, which gets rid of water and keeps the organic biomass, and then ashing the organisms at 480 degrees for 24 hours to obtain skeletal biomass. The carbon value is known to be around 50% of organic tissue and 12% of inorganic. So we were able to work out an amount of carbon stored by each species. First, taking a closer look at the field site for rocky substrata. Cheshire Island was chosen as it is a rocky habitat, but also it allowed for a biodiversity comparison from a study that was conducted at the site in 1998, published in a paper by Barnes and Rockington in 2003. So just on that red arrow at the bottom left, that's the location there for Cheshire Island. 3 transects with multiple quadrats and 3 different depths were sampled for fauna larger than 3 millimeters in size. For this study, we focus only on samples from the 20-meter depth range. As I said, fauna larger than 3 millimeters were collected, which is a challenge especially in cold polar waters with thick diving gloves. Therefore, megafauna was hand collected. Then all other fauna and flora were collected by custom-made Souste suction sampler. So this suction sampler, as you can see on the slide, was designed and made especially for this piece of research. The diagram gives a good overview, but in short, the suction sampler was powered by a 12-volt battery in the boat, and a bilge pump at the back of the suction sampler sucked the water through a hose with a 3 millimeter mesh bag that collected all the animals that were sucked up. So a form of gentle underwater hoovering, if you like. Okay, so this slide gives you an idea of a pre-sampling quadrat on the left and then a post-sampling quadrat on the right, once all the fauna and flora were removed for analysis back in the laboratory. So now taking a closer look at the field sites for the soft sediment work. So the soft sediment stations were the Hanger Cove in the north, where you can see F, E, and D sites, and the South Cove A, B, and C sites. Sampling in soft sediment also used the suction device, and the megafauna were initially collected into a bag, and then the suction sampler was used to suck up everything else within the 0.25 meter squared quadrat. The suction sampler was driven by a 15-litre compressed air cylinder, so a different style of suction sampler, and the Venturi effect lifted all the sediment up the length of pipe and everything was collected in the meshed bag at the end of the pipe. This was also the first biodiversity study for soft sediments that was done in the Antarctic. So just looking at the 20-meter depth, remember, soft substratum assemblages contained 60% less carbon than hard substratum assemblages. If you have a look on this slide, you can see the large amount of carbon stored that was reported within our study. Back in the lab, the animals were identified to lowest taxonomic units. Here are a few examples of the little critters. Clockwise from top left, you have the amphipods, then you have annelid worm, sea cucumber, isopods, chimatia, and a sea urchin. Animals were also weighed and organic and inorganic mass measured. Okay, so quite a bit to take in from this slide, so let's break it down. The x-axis has number of functional groups. The y-axis has amount of carbon. The top graph is organic carbon from tissues, and the bottom graph is skeletal carbon. And the data is for both rocky substratum and soft sediments. The number of functional groups varied from 3 to 12 in each replica quadrat, but both organic and skeletal carbon stocks increased significantly with the number of functional groups, which was the same result that came from the Barnes and Sands 2017 paper when looking at zoo benthic functional diversity at South Georgia. So it shows that the higher the diversity in functional groups, the larger the amount of blue carbon stored within the benthic ecosystems. Okay, so this is all vital for better understanding carbon storage in polar coastal communities. But as I mentioned, Antarctic Peninsula is a hotspot for climate change, and we wanted to try to estimate carbon storage for coastal communities along the whole of the Antarctic Peninsula. By utilizing experts who dive in the Antarctic along the peninsula regularly, we estimated the proportion of dive sites that were known to have rocky or soft substrata. So you can see on this map that was kindly produced by our GIS team at the British Antarctic Survey. And you can see here where we've highlighted hard and soft and mixed substrata along the peninsula. And then we used satellite-derived estimates of ice-free shores on the peninsula that give an estimate of 4,700 kilometers of hard substrata coastline and 960 kilometers of soft. Scaling up and including estimates of macroalgae from the work of Chuck Amsler's group, We estimated 250,000 tonne of carbon at 20-metre depth along the ice-free coastlines of the peninsula. This slide shows another figure from Bax et al. 2021, and we know that ice on the peninsula region is melting. At the time of writing this paper, 54% of the coastline of the peninsula is ice covered, and so blue carbon ecosystems are expected to more than double with continued climate warming. This new habitat will allow new assemblages to develop, drawing down more carbon, providing one of the few negative feedbacks against climate change, and therefore protecting seafloor communities around the Antarctic is expected to help tackle both the biodiversity and climate crisis. This study highlights the need to protect near intact coastal systems and the value of their ecosystem services. This slide shows a proposal for marine protected areas along the Antarctic Peninsula, which are crucial. The paper from Sala et al. 2021, Protecting the Global Ocean for Biodiversity, Food and Climate, showed protection of our natural environments will store more carbon than establishing new or restored habitats from damaged areas. Thank you very much for taking the time out of your busy schedule to listen to my talk on blue carbon storage within benthic coastal communities along the peninsula. Simon and I are more than happy for you to reach out to us should you have any questions or comments, and I leave you with contact information here.
Great, thank you for that, Terry. And I believe Simon is also here. I don't know if it's possible for the IT team to put him into the panelist group so that he and Terry can answer some questions. But that was a really wonderful talk, Terry. Really fascinating. I'm wondering if you've looked at sort of the implications for longer term storage of carbon in these systems, whether there's a difference, say, between the potential for longer term storage between the rocky habitats versus the soft sediments, where you might actually get the carbon being stored for the longer term or not. And it looks like Simon is now a panelist, so both of you can speak as you wish to do so.
Thanks, Alex. And yeah, sorry for the confusion. Maybe should have given you a bit of a heads up that I was going to present today, but luckily we could have the talk shown to everyone. And also nice that Simon can actually join us, because we weren't sure whether that'd be the case. Yeah, on your question, yeah, this blue carbon storage within benthic ecosystems is becoming more and more realized, and the importance, but there is still a lot of work to be done in looking at longer term sequestration, if you like, and especially when comparing the soft sediment habitats and the hard rocky communities, because from our research we showed that actually hard rock communities are storing more blue carbon, but then the potential then to be sequestered longer term, because it ultimately needs to be buried within sediment for that to be sequestered. So there's still a lot more research to be done in this area. So our paper talks more on actual storage rather than sequestration. And actually we need to be a bit careful when we use those terms, because sometimes they're almost being used a little bit incorrectly when people are mentioning sequestration, because sequestration is when it's been completely removed from the carbon cycle and not likely to re-enter. Whereas here we're just actually looking at storage within soft sediments and storage within hard rock communities. So it's a bit of a tricky one to give you a direct answer. Maybe Simon had something to add to what I just mentioned.
Sorry, I missed some of that as I was connecting, but yeah, there is a lot more work to do. And one of the great things, and one of the probably most important things about the rocky communities that we've been looking at around Rothera and in many areas of the Antarctic, is they're actually in close proximity, because these are deep previously glaciated fjords, so they're in close proximity to the sediment. So we would expect quite a lot of that material, the skeletal material, to fall down and actually to collect at the base of those cliffs. So it's a very quick route from where it's growing to the sediment, where we would expect quite a large proportion to be sequestered.
Great, thank you both for those answers. That's really helpful. We do have a question that's come through in the chat as well from Regina Kolzenberg. Says, thank you very much for the interesting talk. I was wondering, when you suggest that the blue carbon storage is increasing with less ice-covered coastlines, did you take the vulnerability of species to thermal changes into consideration and their potential loss?
Yeah, great question. And as with nature, things are always more complicated than you look at. And so we're suggesting here where sea ice is reducing and where glaciers are retreating, so there are newly opened up areas that are ice-free, gives an opportunity for colonization by benthic organisms. So from that respect, then there'll be more zooxanthellae carbon stored within new communities. But of course, there's the added stress in the shallows from things like iceberg scouring. So potentially, the community will be wiped out by a giant iceberg. And of course, then we have the added physiological consequences of thermal tolerance within different organisms. So yes, for example, certain species are definitely more sensitive to increase in sea temperatures, and so their survival longer term will be different to other species. So there's always more questions and a bigger picture to understand the full picture, but here we're just purely talking about opportunities to colonization in new areas when they become ice-free, if that answers the question. Maybe Simon would like to add something as well.
No need to add to that. Perfect answer.
I just have one question, sorry. Thank you, very nice presentation, but I'm concerned, how do you estimate the bottom type, the difference between the soft bottom and the hard bottoms in all the Antarctic Peninsula?
Hi, Carlos. Yes, I did briefly see your question on the right there, but nice to have a conversation. Yes, so we had our data from Rothera on the Antarctic Peninsula, and we have connections with many different collaborators and people who are regularly diving along the Antarctic Peninsula. So they could give us, from the map that I showed towards the end of the presentation, they could give us accurate substrate information from regular dive spots that they use. So we use that information. And then we also then use the GIS team to look at satellite imagery to give us an idea. So it's an estimated amount of what we would suspect would be hard and soft sediments. And here we're merely talking about coastal communities. So we were, from our data, looking at 20 meters depth. And then we went at 40 meters from the coastline out from where the land was to where water was. So it's an estimated figure, but as accurate as we can for the whole of the Antarctic Peninsula.
Yeah, I know you did that because I was in contact with Simon, so he was asking some information, but I was asking if it's possible to extrapolate the data from the satellite. So I'm not sure if it's possible. So this was my question, because I don't know in the other places, but for us, there is no possibility to see anything from the satellite data. It's just that.
Yeah, you're right. Yeah, the satellite data was used from the land perspective, and then the in-water data came from divers that had been in those areas.
Thank you very much.
Okay.
Great. Well, thank you, Terry, and Simon as well. And it's great to see Narissa here now as well, and she'll take over in hosting the rest of the presentations. Thanks, Narissa.
Thank you. I'm so sorry, Terry. I missed your talk. So Luciana Torre is up next, talking about the role of macrobenthic Antarctic community on carbon sequestration in sediments of new ice-free areas. Okay, is that loading?
Sorry,
Luciana, are you there?
But the Luciana has not joined
Yes.
the webinar. Can move on for the
Okay.
next.
Okay, then after Luciana, we have Hannah Oswald talking about, does ice cover impact macroalgal chemical composition in the Western Antarctic Peninsula?
Hi, I submitted a video. Do you have a copy of it?
Scar IT team, do you have a copy of Hannah's talk?
Sorry, we don't have any copy of Hannah Lawson.
Okay, that's fine. I have a copy of the recording myself.
Thank you, Hannah.
We were curious to find if macroalgal composition is impacted by ice cover in the western Antarctic Peninsula. Macroalgae dominate shallow benthic communities along the peninsula, and they play a huge role in nutrient cycling, nutrient retention, and in benthic food webs. Sea ice patterns and duration can vary drastically in small regions along the peninsula. The amount of ice coverage can impact photosynthetic organisms in particular by lowering the amount of light that these organisms can receive throughout the year. However, sea ice is susceptible to the climate. The western peninsula has seen an increase in air and water temperatures over the last several decades, which has led to a decrease in sea ice coverage in this region. To answer our study question, 14 sites were identified based on satellite imagery of annual sea ice duration and placed into either a low, medium, or high ice cover category. Common macroalgal species were collected by scuba divers at each site they occurred, and samples were analyzed for their carbohydrate, lipid, and protein content. For this poster, 4 species were analyzed: 2 browns, Desmorestia menziesii and Hymanothalus grandifolius, and 2 reds, Iridea cordata and Sarcopeltis antarctica. Desmorestia was found to have no significant difference in its carbohydrate or protein content. However, it did have significantly more lipid content in its medium ice cover category compared to low and high ice cover. However, there was not a general trend across ice cover for their lipid content. Hymanothalus and Iridea both had similar results. Both had no significant difference in lipid or carbohydrate content. Hymanothalus had significantly higher protein content in the medium ice cover compared to its low ice cover, and then Iridea had significantly lower protein content in its medium cover compared to the low and high covers. Finally, Sarcopeltis had significantly lower lipid, carbohydrate, and protein content in its lower ice cover category compared to the medium ice cover. In summary, although some individual groups had significant differences in composition, the macroalgae overall had very similar nutritional content across ice cover gradients. This cohesiveness could lend benthic communities nutritional stability when faced with further sea ice loss. And then something interesting to look forward to in the future is we're going to look at other components like storage and pigment to see if those vary by ice cover. But with that, thank you for joining me for this presentation.
Thank you so much, Hannah. We're pretty much exactly on time, so we can probably have a question if there is one. Does anyone have a question for Hannah? I was just wondering, Hannah, with the next stage, what you were going to test for, do you have an expectation of what you might find?
I would expect that we're probably going to find some difference in pigment amounts, but that may be a little bit more correlated to the depths that they were collected at, although I do suspect that they would probably vary a little bit depending on the ice cover that we found, just because you might want more pigments if you're not receiving as much annual light during the year. But again, that may also be related to the depth that we collected them at as well.
Okay. Thanks so much, Hannah. Maybe we can move on to the next speaker. So we have Dolores de Rivas talking about chasing glacial retreat, macroalgal expansion and blue carbon gains in coastal ice-free areas.
Good morning, good afternoon, everybody. Thank you for coming to see this talk, which is Chasing Glacial Retreat, Macroalgal Expansion and Blue Carbon Gains in Coastal Ice-Free Areas. So we wanted to talk about the role of macroalgae in the ecosystems. These organisms are primary producers. They provide habitat, food, and they contribute to the particulate and dissolved matter. So as we know, during the last decades, there has been an increase in the air temperature with consequences in glacial retreat in the Western Antarctic Peninsula. And this has originated newly ice-free areas, more melting of these glaciers, and consequently, due to an increase of sedimentation due to this melting, a decrease in the light penetration in the water column. So we know that macroalgae need hard substrate and good light conditions to grow and settle. And with an increase of sediment input, there is less light penetration, and it has been reported a reduced complexity in the algal assemblages in these areas that are impacted by the glacial retreat and melting. So our question is, are algae not only colonizing but also persisting in these new and highly impacted areas? So in this study, we have two aims to determine the macroalgal colonization patterns in two newly ice-free areas, which are distinguished by different levels of glacial influence, which is a gradient of light availability and turbidity. And the other aim is to estimate the benthic algal expansion, the new biomass and carbon standing stock in areas that have become newly ice-free in recent decades. So our study was performed in Potter Cove, Isla 25 de Mayo, King George Island. And here you can see a gradient of glacial influence which distinguishes three different habitats that were described by Herosh et al. 2018, where we have more glacial influence in the high glacial influence habitat and less glacial influence, more light availability, and more complex macroalgal assemblages in the low glacial influence habitat than in the high glacial influence habitat. So in this area we selected, in this habitat, we selected two areas, which is the low glacial influence area and the high glacial influence area to do our studies. So in the coast of Potter Cove, it is placed Carlin Station. Probably some of you know this station. And below you can see pictures of small creeks that are created in summer or during the months that are warmer, because this is also happening in spring, that when air temperature is higher, above 0 degrees, these creeks are originated and a lot of water, fresh water, goes into the cove with a lot of sediment, and this generates a block in the light penetration that is so important for macroalgae to do photosynthesis. So what we did is a colonization and succession experiment for 4 years. We started in April 2010, putting these tiles, these artificial substrates in the low glacial influence area and the high glacial influence area. 23 tiles were put in each area, and every summer 5 tiles were taken, and we analyzed the identity of the macroalgal species, the richness, and the percent cover. Also, to extrapolate the algal percent cover, dry mass, and organic carbon into all the newly ice-free areas that were originated during the glacial retreat, we did quadrant surveys at 5-meter depths in the low glacial influence area and in the high glacial influence. So these quadrats were 1 meter by 1 meter. We took photographs, and then we calculated the benthic algal percent cover, and with literature we estimated the dry mass and mean organic carbon content. Later, what we did is we calculated the marine area loss of Fourcade Glacier during the time of the experiment and since 1956. So we used the information from the quadrat survey, the data, and we scale up that percent cover, dry mass, and carbon estimations by the total newly exposed substrate suitable for benthic algal colonization, which for us was the probability of hard substrate occurrence based on Herosh et al. 2019. Additionally, we did the measurements of the environmental variables in the water column. So we measured the light, the turbidity, the salinity, and the water temperature. And also we calculated the KD, the light attenuation. So a higher KD means more attenuation due to more turbidity, and a lower KD means the opposite. So our first results show benthic diatoms, Monostroma arioti, Urospora peniciformis, Palmaria decipiens, coralline algae, Adenocystis utricularis, and Desmarestia menziesi. And we saw that in all the tiles, these were colonized by benthic algae, that they differ in the species identity and successional patterns between areas, and that there was a significantly higher richness in the low glacial influence than in the high glacial influence during the last year of the experiment. Here you can see an example of some tiles and how these were colonized by different species during the 4 years of the experiment. In this image, you can see how we calculated how Fourcade Glacier retreated the last since 1956. So all the different colors, especially the violet, is showing how this glacier retreated over time. And then, using the information from the quadrat surveys, the percent cover, and the information we have of all the hard substrate that become available, this is an approximation, a projection of the possible area that could have been colonized by macroalgae due to this glacial retreat. In this figure, we can see the results for KD and turbidity in spring and summer. And this shows that in general, the values for KD, that is light attenuation, and turbidity were higher in the high glacial influence area than in the low glacial influence area. So if we do a conclusion about our results, we know that sediment load is emerging as a key factor determining benthic community structure close to glacial outflows such as Potter Cove. And also that glacial discharge levels can be strongly dependent on air local temperature. No? This is very normal to see high temperatures and a lot of turbidity in the water in Potter Cove. So we saw that the colonization and succession can follow different paths even in similar environments that are in close proximity, principally due to two factors, the light penetration and the sedimentation, that directly affect the benthic algal success and growth. And one of the most important and remarkable results for us was the presence of the Parithia menziesi, which is a late successional taxon that in Potter Cove contributes with the 80% of the total biomass. And we saw an individual growing 2 meters long and with 500 grams of dry mass after 4 years. So this is a very rapid growth. And also a very interesting result is that in the high glacier influence area, there was also a marked colonization, not so much like in the low area, despite these harsh environmental conditions. So macroalgae could colonize and persist over time in these difficult areas for them. So we can say that Glacier Fourcade retreated 1.5 kilometers since 1956, and a lot of new habitat has been exposed that is available for benthic colonization. We also think that this scaling up from local to regional is a very challenging task. So these are our preliminary results, and there are many intrinsic errors, but we know that these are the first steps to do them. So we could estimate that annually the macroalgal community could be expanding 0.012 square kilometers in Potter Cove, and with a dry mass of standing stock of 1.3 tons per year. So this is clear that is enhancing the presence of primary producers over time. So also very important, we know that macroalgae are very abundant and productive in the Western Antarctic Peninsula, but there is very little knowledge about the blue carbon assessment of these organisms in Antarctica. And benthic blue carbon around Antarctica could potentially have a role in mitigating wider CO2 emissions because it is increasing with climate change. So we suggest that monitoring programs could be implemented at a regional scale along the WAP to quantify sequestration of macroalgal carbon. Answering the question we did at the beginning, we know that macroalgae are colonizing and also persisting in these newly ice-free areas. And finally, these macroalgae could be colonizing now and in the future and in higher latitudes. And this generates a significant transformation in the coastal Antarctic ecosystems by increasing the primary production, providing new structures, food and refuge to fauna, and of course potentially increasing the contribution to carbon sinks by adding this new carbon storage in the Antarctic coastal areas. So thank you so much for your attention. Questions are very welcome, and if you want to write your questions or your comments, please see my email below. So thank you very much and have a great SCAR meeting this year.
Thank you so much, Dolores. I see that Dolores is online and we're just about a minute over now. So if there are any questions, do post them to Dolores, but we'll move on to Cesar Martin's talk next, which is on sources and depositional changes of alpha-phatic hydrocarbons recorded in sedimentary cores from Admiralty Bay, South Shetlands. Cesar, are you there, or the IT team? Do you have a poster for Cesar?
Hi, I'm here, but I suppose I uploaded my presentation in the system. Yep. Hi, I think that my presentation is in the system.
Oh, okay. SCAR IT team, do you see Cesar's presentation in the system?
Yes, it's a poster. It's a poster type. We can see that. But we don't have any video from this side.
Okay, let me 1 minute because I have the video here.
You can share the screen from your device.
Let me check.
Dimentichiamo lo snowboard, sarebbe qualcosa.
Hi, I'm Cesar Martins, Associate Professor from Federal University of Paraná, Brazil. I am a member of the Brazilian Antarctic Research Program. Our study is about manufacturing hydrocarbons in 5 sediment cores, short sediment cores in Admiralty Bay, located in the King George Island, Antarctica. We studied the concentration and the distribution of N-alkanes and isoprenoids, pristane and cetane in sediment samples from these 5 cores. These cores represent organic matter deposition around 60 years. Last 60 years, and the sediment cores were dated using cesium and lead 210, and our main results shows variation in the concentration of different N-alkanes. For example, short chains N-alkane that be related to phytoplankton, or medium chain N-alkanes related to prophytes, and long chain N-alkanes related to terrestrial lichens, for example, organic matter. There are some difference between years. That could be related to the increase of temperature in this area around past 50 years, and know the contribution about oil hydrocarbons to the annual alkenes is considered neglected. And I think it's it. Please, if you have more interest or doubt, you can read our paper published in Science of the Total Environment. Thank you very much.
Thank you so much, Cesar. Sorry that there were a few technical issues there, everybody. We might have to just move straight on to Fabio Mendes, who is also presenting a poster. And actually, this is our last talk. It's on photosynthetic performance in forebrown algae. Fabio, are you there, or is the IT team got a poster for Fabio?
No. Fabio has not attended the webinar.
Oh, okay. So unfortunately, it looks like we don't have Fabio's presentation today. So maybe in the last little while, if anyone has any questions for the speakers, that would be a wonderful way to end the session.
Narissa, I had a question for Dolores, actually. If Dolores, are you still online?
Hello.
Hi there. Yeah, I really enjoyed your talk. I was just curious at the towards the end, you were talking about the macroalgae kind of taking over that community as light conditions allow. I'm just wondering if you have a sense as to what they're replacing. You know, do you know what communities were there before the macroalgae were able to start colonizing those ice-free areas?
So you mean in the area that is more in the high impacted area?
Yeah.
Yeah. So before this area was ice cover, and it started being uncovered by 2000, 2003. So we don't know much about the area before that. We don't know if there was something below, maybe fauna. This is something that is being asked by the team that is working with fauna, benthic fauna, but no algae. And once this was uncovered, macroalgae start colonizing this area, which, as I said, is highly impacted with a lot of sediment and turbidity. So there are a few species, but still is an interesting macroalgal assemblage, and that they have colonized and they persist, and we do photo transects and video transects every year, and we find more cover, more species, and we're surprised. But of course, it's not the same that in the area that is not so affected by sedimentation. So yes, yeah. Before, we don't know if there was nothing or fauna, and after it was ice-free, macroalgae colonized in the first from 0 to 10 meters depth mainly.
Okay, thanks, Dolores. That's really interesting. It's amazing that you have this time series of data to be able to calculate just how much that macroalgae is expanding as the glacier is retreating.
Yes, yes. In Carolini, we have been working since more than 30 years. So we have the whole story, and it is very interesting. Yes.
Thanks.
Thank you.
Just following this question, hello, just one more question. You know if there is any predator for this macroalgae with the size enough to impact in these communities? Because most of the predators are very small mollusks. Since there is not too much fish there, do you know any kind of predator for this macroalgae?
No, there is no any predator for this macroalgae. They have different defense systems, and no, they are not. It could happen a little bit, but no, there are no predators that could impact. These communities are impacted by the environmental variables, no? The sedimentation, the lack of light, ice coring. These are the changes in sea ice cover. This is what is affecting most rapidly.
Dolores, I was just curious, it may have been mentioned already, but you said something about how this is done every year. So you're expecting to be able to do this every year into the near future as well?
Yes. So we did the experiment, the colonization experiment, and the calculations, the approximations of how much these assemblages could have expanded during this time. And then what we do in different newly ice-free areas, here I showed the result for 2, but we have different that were uncovered different years. So we do photographs and video transects, and we continue monitoring the environmental variables to see the changes over time.
That's amazing.
Yeah, it is, as I told you, the main effect we think is sedimentation and the turbidity, because if you go in summer, the water is brown, directly brown, but there are other important factors that we are studying, for example, ice coring. So yes, we are following these changes every year. And of course, this is very local, so our next step is to compare with other bays and coves around the island and in other parts of the Antarctic Peninsula.
Oh, that's great.
Yeah.
So I look forward to following it more, Dolores.
Yeah, thank you so much. Now we are participating in a project that is called Coast Guard. So it's different countries along the Western Antarctic Peninsula. So there we have different points to compare, and it's going to make all this work more regional and more interesting to compare with different latitudes.
I might see you then at Coast Guard in Chile in October. Yeah. Yeah, that would be amazing.
Yeah. We are planning that.
Yeah.
Yes. See you there. And probably more people that is in this session.
Yeah, hopefully.
Yes. Okay, thank you.
Well, oh, thank you. We are probably at the end of our session, and unless anybody else has any questions before they close us off. Okay. Well, I just wanted to say thank you to everybody, especially Alix Post, and sincere apologies for coming in late to the session. It was wonderful to see the talks that I did, and I'm going to go back and look at everybody else's online. And yeah, thank you to the SCAR team, the SCAR IT team, and I hope everyone enjoys the rest of the conference.
Thank you very much.
Thank you, everyone.
Thanks, Narissa.
Thank you, everybody.
Thank you very much. Very nice session and talks.